Hello guys, so welcome back to my channel. So today we are going to study about streak plate technique. So what is streak plate? Imagine you have a yummy pizza with lots of toppings and you want to separate them into slices. That kind of like what the streak plate technique does in microbiology. It's a way scientists separates microbes from a mix so they can grow and study them individually. Want to learn more about the microbes? Comment below. Let's get started. So in the streak plate technique, you're gonna take special plate with agar for microbes to grow on. Then use a sterile loop to spread a tiny drop of microbes mix on the plate. Streak the loop across the plate in a pattern spreading microbe. Then incubate the plate and let microbes grow in colonies. But guess what? There are different methods to streak these tiny creatures on the agar plate. Let's discuss different methods to spread microbes, that is, different types of streaks. Okay, so here are four types of a streak plates. First is a quadrant streak, second T streak, third continuous streak, or also we call it as a zigzag streak, fourth radiant streak. So quadrant streak is something that is most commonly used. Okay, so in the quadrant streaking we are going to divide the agar plate into four equal sectors, 1 2 3 4. So each one is a different sector. So here we are going to discuss about how quadrant streak is performed. So first, before performing the quadrant streak, we have to sterilize the inoculation loop. After sterilization of inoculation loop, here we are going to take a agar plate and streak it in the first quadrant. So as you can see in the first quadrant, the number of microbes will be more. It means it has a high concentration of microbes. Then second quadrant and third quadrant respectively has a diluted form of microbes. This is the second streak. See, after performing the first streak, we are going to sterilize the loop. Then again we are going to use for the second streak. Then after performing the second streak, again we are going to sterilize the inoculation loop, and then we are going to perform the third streak. Okay, so later we are going to do the fourth streak. In the fourth streak, we have to streak in such a way that single species can be obtained. Understood? So this quadrant streak is mainly used to obtain the single species. Understood? So our next method is continuous streaking. In continuous streaking, the sample is spread across whole plate, either in a zigzag way or a continuous streak. But the thing is, here we are not lifting the loop once again. Okay, in the quadrant streak what we had performed, right? So in that each time we were sterilizing the loop, but in continuous streak we should not sterilize the loop. Before performing the continuous streak, once we have to sterilize the loop, then we have to take a culture and spread the culture in a continuous way without lifting the loop. Okay, so this method is good for antibiotic testing or culture of lots of bacteria. As it is a simple technique, the bulk growth of microbes can be obtained. Our next streak is the T streak. So here Petri plate is divided into three sectors. First streak across by swiping loop full of sample across 1/3 of a plate. Then turn the streak. Next sector touching previous streak once. Okay, and finally the last streak by sterilizing the loop and touching the last sector once. This method helps to separate the bacteria and quickly and easily. It is very easy to perform. Okay, so our next method of streaking is a radiant streaking. Radiant streaking is a technique when you streak outwards from a central point like a spokes on wheel. See, in this you can see it is a central part and all the lines you can draw as a spokes that you can see on a wheel. Yes. So here place a loop full of sample in the center, means here you have to place a loop full of culture, and then you are going to streak the line in such a manner the streak should be outside from a central point. This method is to isolate bacteria from a concentrated sample. Spreading bacteria out makes the isolation easier. So in the concentration, the center, the microbes will be more, right? So when you streak out towards here, you can in this points you can get the colonies easily. So it makes easier for the isolation of one type of organism. So here is differentiation between the all four types of streak that we have studied. First is the quadrant streaking, that is to isolate. The purpose of quadrant streak is to isolate individual colonies. So plate is divided into four sections and we are going to sterilize the loop between each of the streak. Next is our continuous streak. It is the purpose of continuous streak is to achieve heavy bacterial growth, that too in a zigzag pattern we are doing, and in between no sterilization of the loop. T-streak. For quick isolation of colonies, this T-streak is performed. We are going to streak in a T shape and we are going to sterilize the loop in between each of the streaks. Last is the radiant streak. The radiant streak is to isolate bacteria from a concentrated sample. Streak outwards from a central point that are like spokes. Okay. So here are few questions. Try to answer those. Which streaking technique is best for isolating individual bacterial colonies? Is quadrant streaking. Which streaking method is used for achieving heavy bacterial growth? Correct option is continuous streak. In which streaking technique do you streak outwards from a central point? Radiant streaking. Which streaking technique involves streaking in a T shape? Answer is T-streak. What is primary purpose of sterilizing the loop between the streaks? To prevent the contamination. Which of the following is benefit of the quadrant streak method? Isolate individual colonies. Hope you have liked this video. So, thanks for watching. Did you answer all the questions right? What is your marks? Comment below. Do subscribe to our channel. Thank you.